Hi, class. This unit is on microbiology. So we are going to have 3 lessons. First lesson will be on bacteria. The second lesson will be on fungus and protists. And the last lesson will be on viruses. But before we get into different types of microorganisms, we are going to talk about the word micro and what microbiology is. So, micro, the word means small. So, microbiology is a field of biology that studies organisms that are too small to be seen with the naked eye. Some examples include algae, bacteria, mold, and yeast. And we're going to talk about all of those different types of microorganisms. Now, in this course, we are not going to do um, any labs, right? Because number one, it's an online course, and number two, uh, it can be dangerous to do a microbiology lab. So I want to talk a little bit about special safety measures that have to be taken when we do microbiology labs. So some microorganisms we know can cause diseases in humans. They're pathogenic. So when we collect and work with microorganisms, we have to take some precautions to um, to prevent ourselves from getting ill. Okay, so I'm just going to talk about a few little techniques and a few little things that uh, we do in microbiology that we don't necessarily do in with other types of biology. So first, we're going to talk about how we collect, grow, and observe microorganisms. So, um, a sample of a microorganism or a sample of a living thing is called a specimen. And we can collect microorganisms from a lot of different places. We can collect them from the soil, we can collect them from our water, we can collect them from food, and we can collect them from bodily fluids, such as blood and urine. We like microorganisms to be in the soil usually because they have special relationships with plants to help them grow. So usually when we check the soil, we're checking to see if the correct microorganisms are there. Water, we don't like to see any microorganisms in, especially our drinking water. So there's a job where you can check water to see if you have any um, disease-causing organisms like E. coli. And then bodily fluids, we also don't usually want to see microorganisms in your blood or your urine because that usually means you have a disease. Okay. Inoculation and incubation. So when you get a sample of a microorganism, there might not be very many of them there, which makes them difficult to study. So what we do usually is we find something, an environment that is um, that the organisms like to grow in. So these would be things like nutrient-rich liquids or jelly-like solids. Um, and we introduce the microorganisms to these materials so that they can reproduce and we can more easily look at the type of microorganism that we found because there's more of them. So the process of introducing microbes to an environment that they can easily grow in is called inoculation. Um, and there is a special apparatus called an incubator that provides the right temperatures for microbes to grow. A lot of the disease-causing organisms, they grow at human body temperature. So usually when we study microorganisms, they like a little bit of a warmer environment. So if we incubate them, Okay, if we put them in an incubator, it's like a little room where the temperature is hotter, or we can control the temperature at least and set it to a temperature where the microorganisms want to grow. So after we inoculate and incubate the organisms, there are more of them there, and we can inspect them. So when isolated populations of specific kinds of microorganisms have been grown, their physical properties are inspected and analyzed. Since most microbes are between 100 micrometers and 1 millimeter in size, they're too small to see with the unaided eye, so we use microscopes um, to inspect each individual microorganism up close. We can also use plates. So this is a petri dish here. It's a shallow glass dish that we can put some nutrient jelly in, and then we can inspect groups of microorganisms called colonies, right? And sometimes you can tell what microorganism it is, depending on like the shape and the color of the colonies. But usually we also inspect them under uh, the microscope. Now, um, what's really, really important, because we can't really see these microorganisms, is we work in a sterile environment, which means that we clean everything really, really well. And sometimes we heat things very, very hot to sterilize them so that we don't contaminate uh, one sample with another sample. So we clean our tools really, really well, and we tend to use disposable tools that we can use once and then throw away, so that microorganisms don't accidentally get into our experiments. And then now we're going to talk about safety. So some careers that deal with microbiology also deal with bacteria that are infectious, right? Disease causing, contagious to humans and other organisms. Therefore, care must be taken to avoid touching or breathing in microorganisms. So we usually wear, if we do microbiology, um, extra protective equipment, including face masks, gloves, lab coats, and even hazardous material suits. Sometimes they shorten the name of hazardous material suits to hazmat suits. And finally, we're going to talk about careers in microbiology. There are a lot of careers in microbiology. So microbiologists don't all work in labs. Other careers in microbiology include farmers, right? So you can study blight, which is a fungal disease in potatoes, for example, and water treatment technicians who keep algae and bacteria from our water supply. So you might work on a farm, or you might go between a series of farms, or you might go between different neighborhoods and collect water samples from different places. And that can be a job where you can work outside. So now we're going to talk about some what makes bacteria different from other types of cells, because bacteria uh, belong to a type of organisms called prokaryotes. So we have to learn about the difference between prokaryotic and eukaryotic cells. Um, I think what I'm going to do is I'm going to play just the beginning of this Amoeba Sisters video. It's a long video, but I want to make sure that you have the correct, that you can hear this video. Okay. Here we go. So we're going to learn about the difference between prokaryotes and eukaryotes.
If you had to think back about the most exciting day you ever had in a science classroom, which day would that be? Now, looking back through the years, we have a few. The time we participated in an earthworm dissection, the time we took apart an owl pellet, the osmosis eggs, all of the fruit flies and genetic experiments. Oh, I could go on, but I will never forget one day in my ninth grade science class. My teacher brought in pond water, and I put one drop of pond water on a microscope slide and saw the most amazing thing ever. I saw an amoeba, a single celled amoeba on that microscope slide, and I was forever stuck on science from that point on. Because I could not believe that this little cell was there, alive on this slide, still eating because that's what amoebas do a lot. But to imagine that every person is actually made of billions of cells. Of course, not amoeba cells, but animal cells, billions of animal cells. That's fascinating. In fact, it really makes you reflect on some of the incredible statements of the modern cell theory. The modern cell theory includes the following. First, that the cell is the smallest living unit in all organisms. And second, that all living things are made of cells, one or more cells. The amoeba I observed was a single celled organism, so unicellular. Humans are made of many cells, so multicellular. And third, all cells come from other pre existing cells. Cells have their own little world inside them. They carry genetic information, they can divide, many have functions and processes that their organelles, structures inside them, can take care of. On our planet, we can divide cells into two major groups. As a cell, you're either a prokaryote or a eukaryote. Bacteria and archaea are prokaryotes, but everything else, plants, animals, fungi, protists, are eukaryotes. Both prokaryotes and eukaryotes have genetic material. Both have cytoplasm. Both have ribosomes, which are small organelles that make protein. Both have cell membranes, which control what goes in and out of the cell. But what makes them different is a big deal. Prokaryote, the pro rhymes with no, and they have no nucleus, which holds the genetic material and controls the cell's activities. Prokaryotes have no membrane bound organelles. Membrane bound organelles are fancy organelles like the nucleus and mitochondria and Golgi apparatus. Eukaryotes, the U rhymes with do, they do have membrane bound organelles. So now you may be wondering, well, what do the organelles do? What are their functions?
We already learned what the organelles do, at least the eukaryote ones in our first unit for plant and animal cells. So we are not going to relearn about them. There we go. Okay. What we are going to do is we're going to take another look at the differences between prokaryotic and eukaryotic cells. We are going to look at their morphology. Morphology is a word that means the shape and the structural features of an organism. So we're going to look at how prokaryotes and eukaryotes look different, how their cells look different. So here we have a diagram that shows a eukaryote and a prokaryote cell. The prokaryote is on the left, and you can see that the prokaryote has a chromosome, right, which is DNA. But you'll notice that the DNA is not in a nucleus, right? Here in the eukaryotic cell, the nucleus contains the DNA, right? But for the prokaryote, it's floating just in the cytoplasm by itself. So in prokaryotes, you can't see the dot, like the big dark structure called the nucleus. It's the chromosome is just sort of floating around. Both prokaryotes and eukaryotes have a cell membrane. They have a they have ribosomes. They both have cytoplasm. They can both have organelles like flagella that help them to move. But prokaryotic cells do not have membrane-bound organelles. Okay, so you don't see, for example, a Golgi body in the prokaryote side. So prokaryotic cells don't have a nuclear membrane, which means they don't have a traditional nucleus. They do have the DNA that usually goes inside the nucleus, but they don't have a nucleus. And they don't have any other membrane bound organelles. So they can seem kind of empty compared to eukaryotic cells. Eukaryotic cells do have a nuclear membrane and they do have membrane bound organelles like the endoplasmic reticulum, Golgi body, vacuoles, mitochondria, and chloroplasts. Both contain cytoplasm, ribosomes, and um, chromosomes. Now, in this unit, we may talk a little bit about organisms by their scientific name. Okay, every organism that has been discovered on Earth is given a scientific name, and the reason why that is because scientists, no matter what language they speak, they need to be able to talk to one another. Um, so when we name organisms, we name them by um, the way they are classified. So we have, for example, different kingdoms of organisms like plants and animals and fungus and bacteria, right? And we further divide these categories, these kingdoms, into smaller and smaller categories, right? So you can have an animal that happens to be a cat, right? So, they would be in the group like Felidae, right? Which is the cat group. Um, and we eventually get down to the smallest two categories called the genus and the species. So every species has its own species name, and then sometimes they're grouped with very, very closely related other animals in a genus. Okay. When we write scientific names, um, we in order to write them properly, to type them, we use an italic font. When you're in your word processor, there's the B for bold, the I that's sort of on a slant, that's for italics, and the U is for underline. So scientific names should be, you should type them in italics. The genus comes first, and it is capitalized, and the species name comes second, and it starts with a lowercase letter. Many microorganisms are known by their scientific names. So humans are called Homo sapiens. Homo is the genus, and sapiens is the species. There's also Escherichia coli, which we call E. coli for short, and Escherichia is the genus, and coli is the species. There's also Canis familiaris, which is our domesticated dogs. Canis is the genus, and familiaris is the species. Okay, so we could write the scientific name for a horse. The genus for a horse is Equus, and the species is Cabalis. So we would write the genus name first, Equus, and we would make sure that it has a capital letter. And we would write the species second, and we would make sure the species starts with a lowercase letter. Okay, we could also write the scientific name for zebra. Zebra is the same genus as a horse. It's still Equus, but its species name is different. It's Equus burchellii, right? Species are often named after the scientists who discovered them or who named them, right? So here we have Equus burchellii as the name of the zebra. And we can tell that horses and zebras are very closely related because they have the same genus. Now, so we talked a little bit about kingdoms. Kingdoms are large groups of of organisms based based on traits that they have, based on characteristics they have. So many different living things can be found in microbiology labs. There are microscopic animals, plants, fungi, protists, bacteria, and archaea. So how do we tell the difference? We have a little chart that has some of their characteristics. So archaea and bacteria are both prokaryotes. So if you see a cell that doesn't have a nucleus, it probably is an archaea or a bacteria. All archaea and bacteria are single-celled organisms, right? Collectively, sometimes we refer both to both archaea and bacteria as bacteria. Sometimes we call archaea archaebacteria. And they're very similar organisms because both of them don't have organelles and both of them are single-celled, but there are some differences. Um, both archaea and bacteria can make their own food or consume other organisms, but archaea can live in extreme conditions. And we'll talk about that when we talk about archaea. And their cell wall lacks biomolecules that are present in bacteria. So the difference between archaea and bacteria is their cell walls have different molecules in them, and archaea can live in harsh environments. All archaea are microorganisms, meaning we can't see the complete organism unless we use a microscope. Bacteria are different again because of the cell wall consists of different biomolecules than archaea. But other than, and they don't live in extreme environments, but other than that, archaea and bacteria are very similar. We also have protists and fungi. Protists and fungi are funny in that they are both eukaryotic and they both can either be single-celled or multi-celled. Okay, so for example, protists include algae. So algae can be sometimes single-celled, sometimes multi-celled, and funguses can be mushrooms, which are multi-celled, or mold and yeast, which are single-celled. Um, Protists are interesting because they are a little bit like archaea and bacteria, and some make their own food, some consume other organisms, and some do both. But one thing that makes protists different than um, other single-celled organisms is that they can move. So protists are basically where we throw things we don't know what to do with. If it's like a fungi but it can move on its own, right? We'll call it a protist. If it's like a plant but it's single-celled and it can move on its own, we call it a protist. Fungi, all funguses. So funguses are different than plants. A lot of people think a mushroom is a plant. A mushroom is not a plant because it's not green and it can't do photosynthesis. So fungi are not movable, like they don't move like animals, but they don't do photosynthesis like plants. They can be single-celled or multicellular, but they always consume other things for food, usually dead things, but they can't move like animals. Plants are multicellular and eukaryotic. They have a nucleus. They consist of more than one cell. They all make their own food. Animals are eukaryotic. They have a nucleus. They're multicellular and they can move. And all animals consume other organisms for food. So we don't have any photosynthetic animals yet. So we have some practice questions. You're looking at a single-celled organism under a microscope. What types, what kingdoms of organism could it be? So it could be an archaea, it could be a bacteria, it could be a protist, or it could be a fungi. It could not be a plant or animal because all of them are multicellular. Okay. Um, Fungi and protists we know could be both, so we have to include them. You are looking at a multicelled organism that consumes other organisms for food. What types of organism could it be? So in this case, both plants and animals are multicellular, but animals are the only ones that consume other organisms. So in this case, you must be looking at an animal because that's the only one that has both of the characteristics. And the last question, you are looking at a single-celled organism that makes its own food. What types or kingdoms of organism could it be? So in this case, it could be an archaea, a bacteria, or a protist, but not a fungus. Pretty cool, right? So we can use those different characteristics to tell one organism from one type of organism from another type of organism, right? But it can be a little tricky, and sometimes we have to do a little bit more research to narrow it down to one type of organism. Okay, so now we're going to look at bacteria and archaea, right? So bacteria and archaea, we said were prokaryotes. Many prokaryotes have some of their DNA in a small circular structure called a plasmid. So they still have chromosomes, but they have little extra pieces of DNA, and you can see some here, right? These rings are called plasmids, and they're extra DNA that don't always get replicated when they have children. And the other thing is that bacteria can share them with each other. So they can, I think I want to start this. This should be its own little thing. Okay.